Hello everyone, I am Laurent Turpin. I am a second year PhD student from Lyon in France, and I will present you a work done with Thierry Gautier and Jonathan Rouzaud-Cornabas. It's called X-Aevol, a GPU implementation of an evolutionary experimentation simulator. So this work relates to evolutionary biology, but I am a computer scientist, so this is my vision of the domain. Evolutionary biology is about studying the oldest genetic algorithm ever. The problem is that nobody has information on how it works. You might already know it. This genetic algorithm is known as life. Because of all these unknowns, researchers are trying to retro-engineer life to build its user manual. To do so, there are different ways. In vivo observation, like Darwin or like paleontologists with fossils and analyzing the DNA, it means the genome of the organisms. You can also do in vitro experimentations, which is similar to in vivo but using controlled environment and population. A famous example is the long-term evolution experiment of Richard Lenski that observed E. coli bacteria evolve for 30 years. And yes, the problem of these methods is that they need lots of time, effort, and money. So, as many other things in the world, we can use computers to reduce the needed time, effort, and money. Biologists can then study evolution through in silico experiments. It works as in vitro experiments with a notable difference. There is no implementation of life on computers. So you have to create your own implementation, which will be a model of life. This model will be a genetic algorithm, but it must be convincing enough for biologists and ideally run faster than life, because you don't want to wait years to have the desired result. I have to insist on the purpose of the model. Of the genetic algorithm that we are studying. It is not a tool to solve problem like an optimization problem. It is a model to study the evolution. So the model, the output data, and the usage is constrained by this purpose. We want to understand evolution, not use it. In other words, we do not care about the solution. We care about the path taken from the start to the end and what are the mechanisms that lead to take this path. The model that we are using is EAHPC. It is a model inspired by the biology of bacteria and mimic features observed on bacteria. The important features are the genome and the mutations. The genome is circular, and the process to get the fitness of an individual from its genome is a bit complex. It involves the identification of segments inside the genome called RNAs that you can see in blue on the diagram, and then segments inside these RNAs called genes, the orange sequence on the diagram. And finally, reading these genes to eventually get the fitness. For the variation of the genome, it only occurs through mutations that can take the form of point mutations that change a little bit of the genome, or rearrangements that can duplicate the whole genomes or cut the half of it. You can understand that the model implies irregularities inside a genome and between individuals that will have different genome size. Indeed, once the model has been transformed into an algorithm, these irregularities are much visible. On the figure at the right, you can see a glimpse of the algorithm that implements the model of EAHPC. The part that caught our attention is the evaluation process of an individual. That means the process that gives the fitness from the genome. It is indeed the most time-consuming part of the algorithm, and it is where all the irregularities lie. These irregularities are due to the complex structure of the genome. The genome contains RNAs that contain themselves genes, and their number are not known in advance. And this complexity is a problem if you want to run the program faster, because it is a subject of interest. For now, the actual CPU implementation computes millions of generations with thousands of individuals in a few days. But we want to simulate millions of individuals, so it would take thousands of days That means years, and it is not reasonable. Remember, the program should run faster than life. The response to that problem is X-Aevol, the GPU implementation of Evol. But what does it mean to use a GPU? Well, let's take a simple task, cooking French fries. You have to cut the potatoes, so from one potato you have to get even strips. First, let's say you are using a knife. It is a sequential process. You do one cut after the other. It's like a CPU. On the other hand, let's imagine you are using this amazing device, a potato chipper. It has several blades arranged in a grid and can cut all the strips in one action, in parallel. It is like a GPU with the SIMD programming model, single instruction, multiple data. It is where the power of GPU lies, doing lots of similar things in a massively parallel manner. However, what happens if things are unbalanced, irregular, when you have particular cases? Well, you have to deal with that. In the case of Evol, we found multiple sources of irregularities and solved them thanks to different solutions. We had to store data without knowing their number in advance, so it led us to allocate memory early. The access of data structures is done with concurrency, and atomic operations help us to solve this problem. At last, the nested nature of data, remember the genes inside RNAs inside the genome, led us to modify the Bayes algorithm, and we used specific parallel algorithm to fully use the GPU capabilities. At the end, here are our performance results. The diagram on the left shows the speedup using NVidia A100 GPU against CPL execution, depending on the genome size, with increasing population size. The computation is to evaluate n times a single genome, n being the population size. We see that with a large enough population size, we reach a speedup of 1000. The fact that using less individuals lowers the speedup suggests that the GPU is not saturated enough and that the memory loadings are not completely overlapped by arithmetic computations. Indeed, this program is mostly memory bounded. Nonetheless, X-Aevol still yield high speedup with more usual population size of 1024. We also tried to compare the performance between two generations of GPU architecture, the V100 against the A100. The speedup in red on the diagram is increased by 50% with large enough population using the A100, even though the new architecture has only 35% more computing capability. In fact, the more recent architecture has more memory capacity as well, with higher bandwidth, larger cache, and so on. A last test is about a larger scale irregularity. I told you that genome size varies between individuals because of the mutations creating an heterogeneous population. The differences in genome size imply differences in computation time that create work imbalance. The bottom diagram shows the repartition of computation time among four heterogeneous populations, represented with four colors. Although there is a peak of representation, some individuals can take twice or three times as much computation time than others. This imbalance creates a scheduling problem that is, for the moment, handled by the NVidia runtime. The diagram on the right shows three comparisons between a clonal population, when you evaluate one genome n times, and an heterogeneous population, when you evaluate n genomes once. The heterogeneous population take a little bit more time to evaluate, and this effect is accentuated with a lower population. This can be expected because the list scheduling technique used by the runtime loses efficiency when the number of tasks is too low. At this point, we should summarize what we learned so far when dealing with irregular programs on a GPU. For memory allocation, you certainly have to do it in advance using an estimation of what you are going to need. We do not recommend the usage of dynamic allocation because of their poor performance on GPUs. Do not hesitate to rewrite your algorithm to leverage parallelism. In a nutshell, adding work for the sake of parallelism can be great. And finally, use what the hardware can offer to you, for example, atomic operations. To conclude, porting an irregular algorithm on GPU is not straightforward. That being said, we did manage to greatly accelerate Evol with tweaks in the algorithm, and we can hope doing experiments with millions individual in a few days instead of years. However, we have still ongoing work. We have to complete the implementation of the genetic algorithm, and last but not least, we noticed that millions of individuals do not fit in the memory of the GPU, which is around tens of gigabytes. We then have to use multiple GPUs. That means handle the work balance and data distribution between those GPUs. So thank you for your attention and bon appétit.